Hello everyone. So today I'm going to teach you how to use your newly generated DNA sequence. So let's start. So these are some newly generated DNA sequences that I'll be using for today's lesson. So let's start. So in order to first use your sequence, you have to know if your sequence is usable or not. So you have to check the quality of your sequence. So how to do that? So in order to do that, you have to open this ABI-AB1 file, and you can see here this peak shows your sequence. Okay, if this peak indicates your DNA sequence, and so normally it should be only single at one region. It should be single peak, but you can see here this region, the multiple blue, red, and multiple lines are here. So this means that this is not a very good sequence in this region. And further you go, you can see it is going down and down and down, and so this is not a very good sequence. So this kind of sequence, we are not supposed to use this kind of sequence, and we will never use this kind of sequence. So I'm not going to use this sequence for our further study. You can see here some overlaps are here, but other than that it looks good. Little overlap also here, and okay, little bit there is overlaps here and there, but not so much. You can see it's kind of usable sequence, but we will also not use this sequence for scientific studies. We can use this for identification purpose, but for scientific studies like phylogenetic studies, we will never use this kind of sequence, so we will also not use this one. And then we have this sequence, totally bad sequence. Never use this one, just close it. And another sequence we have here, you can see, so this is the sign of a good sequence, but here also you can see some overlaps. Other than that, it looks good. So what to do with these overlaps, and what are we supposed to do if our whole sequence is good, but you will find overlap in the middle, and you will find overlap at the end, toward the end. So if you find some overlaps toward the end like this one, you just have to remove from here. But if the overlaps are in the middle, then keep it. If like you have only one overlap in the whole sequence, if there is only one overlap in the central region, so you can exclude, sorry, you have to include that one, not exclude. And you can see this sequence looks very good as compared to others, other than the region here, this overlap. So what we're going to do, so we will remove this region, and also you will find some region like this one. This is okay, it's not bad, because this is the region of your initial sequencing region. So we will also remove this one. It's okay if we see this kind of sequence here, but we have to remove it. Also at the end you will find something like, okay, it's not here, but you will usually find those wobbly region like this one at the other end also. So now we're going, so in order to use this sequence, we will remove this region. So how to do that? So you can see that this region comes under 80, or you can say 85. So from 80, 81. Okay, so we will remove from 81. So go to edit, and then 81, and then delete. So how did I did that? I will show you again. You just have to go edit mode, and then click here, shift, then click here. can delete this now. Just press delete and it's done. So now what we're going to do with this, we have to save this one as, let's see, folder here, 241, and just 241 ID is 1. Save it. Then we can close this one. So in this case the ITS1 is good, but the ITS4 you can see it's really bad. So what are we going to do with this? We are never going to use this one. So I have another sequence for 241. So you have to resequence that region. So this is the other ITS4 that I have generated again. So if you get bad sequence, you have to do it again. You can see this one is better than before. It shows some overlaps like here, but overall it looks kind of good. So we will also remove this initial region, wobbly region from 90. We will remove it. So this is the case of, you know, not very good sequence, but I'll show you what to do with not very good sequence. 241 DS4. So now we have both of these sequences, that is ITS1 and ITS4. So what are we going to do with these two? We're going to use both of these in the phylogenetic analysis? No. I'll show you. In order to do the correct way of analysis, you have to first open, okay, I'll show you. So first open this ITS1. We know that ITS1 is forward sequence, so we will keep this as it is. In this we will import another sequence file alignment, that is our ITS4, and we know ITS4 is reverse complementary to this ITS1 sequence because this is reverse sequence. So in order to complement both of this, we have to press Ctrl Shift and R, so it will do the reverse complement. So now we can select both of these sequence and align them. Okay, you can see it has, the alignment is done. You can see this properly aligned, but we are not done yet. So in order to know if your sequence is good or bad, you have to create consensus sequence. You can see here, if your sequence is good, then you will never see letters other than ATGC, that is our normal DNA sequences. But if your sequence is bad, then you will see some letters other than ATGC. You can see here, because they are not complementary. So this is what we found, the letter S. So I don't know what this letters S indicate, but I think it's kind of like mutation. Maybe it shows the mutation, or maybe a protein sequence, I don't know. But you have to keep this, don't delete or manipulate this one, just keep it as it is, so we can know what kind of changes mutation has occurred in your sequence. So we will manually observe more sequences, more sequence like this, more letter like this one, and also here we have, so like this. So now that we have created the consensus sequence, we have to copy this one, copy, Ctrl C, and then create new text document and then paste it here. So this is our the main usable form of DNA sequence. This is ITS region. So this is the combined ITS region of the species 241 using the forward and reverse sequence. So this is what we are going to use. So you can now save as 241 ITS region dot FASTA. So this is what we're going to use, and you will see that I have kept the letters S and other letters other than ATGC. It's okay, don't remove those, just let them be there like this. So we can use this one for our phylogenetic analysis or other kind of analysis. So this is a case of not very average kind of DNA sequence. So now I'm going to show you the good sequences, how they look like. So for example, this is a good sequence. You can see very good peak, no overlap. You can see the initial phase of the sequencer and no wobbly, no overlap. You will never find overlap. This is a very good sequence. This is how a very good sequence looks like. Although some of the peaks are very high, but it's okay. It is a good sequence. Just because it is high doesn't mean it is bad. It is actually a good sequence. And you can see at the other end you will find this wobbly region, kind of like that. So we have to remove this and this initial phase. So we can do that by just looking at here. We will remove from 920. So why am I removing the end region first? Because if we do the initial region first, then this will overlap, replace the region here or the region that I have deleted at the initial phase. Like for example, if I delete from here to here first, then you will not find the 920 region. Okay, so that's why we have to first remove the end and then the initial region or the start region from 21. From here we can remove this one and then save as so this is the forward sequence, and then we have our reverse sequence here. You can see the bubbly region, and this is actually a normal sequence, the perfect normal sequence. You can see even the peaks are not that high, the average peak level. This is some of the peaks are high, but it's okay. You can see at the end another wobbly region. It means this indicate that your sequence has gone through the initial and up to the final process. So we can remove from this 920 or 930 also. So we can go to the end. From 930 we will remove, because these are not our sequence, not our DNA sequence. And from initial phase we can remove from 15. Delete. Now save this 251. Like that we will again do the same thing, and you will see, now you will see that difference between good sequence and bad sequence. import here, and we can do the same, Control Shift R to reverse complement, and then select all, then cluster multiply alignment, run. Okay, now you will see that you will, okay, let me do the create consensus sequence. Now you will see that you will, if you manually check, you will never find other letter other than ATGC, because this is a good sequence. You will not find that. This is a very good sequence, so you will not see those letters other than ATGC. You can see. So this is the sign of a very good sequence. When you create the consensus sequence, you will find that there is no overlap. You can see no letters other than your normal ATGC. Now we can save this one, and this is very good sequence, so we can use it for our phylogenetic analysis. So this is an LSU region, so 251 and LSU. Okay, no, so we are going to, sorry, this, not, we are not saving, we are supposed to copy this one, copy, and then create text document, and then paste 251 and LSU region. Okay, so now we are done with our sorry, so this sequence is very good sequence. We can use this one in our other analysis like phylogenetic and alignment. So now you can go to, now you can blast search the result. So it will show the most so you can see my sequence is 100% similar to Phascoporeus senex and Felinus reververinus. That is also Phascoporeus reververinus. You can see 100% identity. Even Quadricore is 95%, but the percentage identity is 100%. So this is my species that I have newly generated. And also you can do another blast. Also we'll do another blast for the somewhat mid-range good sequence, that is our 241. So this is the ITS region. We'll do blast. So you can see it is very similar to uncultured fungus clone and Tricheptum bisogenum. So this is actually my this species is actually my Tricheptum bisogenus genome. My this one mid range sequence is also very good sequence, so it it shows something somewhat similar, and I know what it was. So it is actually Trichepton bisogenum, and it is showing 99% similarities, so that's a good sign. So this is it for today. We'll see you in the next lesson. Thanks for watching.